This video will discuss characteristics of mitochondrial DNA and processes associated with inheritance of mitochondrial DNA. By providing important background information on the mitochondrial genome, we hope to equip you with the tools you need to tackle the questions that will be addressed in the video and beyond that pertain to mitochondrial DNA.
Scientists have hypothesized that the mitochondria and chloroplasts were both once free-living prokaryotic cells, but ended up inside a host cell either by being phagocytized or by parasitizing the host cell. The mitochondria and chloroplasts survived as inner cells that made energy for the host cell to use. Over time, the organelle and host cell evolved together, and now they function as a single organism. This theory is called the Endosymbiont Theory, and there is a lot of evidence that supports its claims. Such evidence includes that chloroplasts and mitochondrial DNA are circular, just like prokaryotic DNA. Additionally, mitochondria and chloroplasts are double membrane-bound organelles, which is characteristic of a cell that has been engulfed. Also, in terms of evolution, there are several instances of modern protists that host endosymbiont bacteria. Furthermore, the ribosomes of chloroplasts and mitochondria are structurally similar to eubacteria ribosomes. And lastly, antibiotics that do not affect protein synthesis in eukaryotes have been shown to inhibit protein synthesis in eubacteria, chloroplasts, and mitochondria.
Mitochondria are ATP-generating organelles that range from about 0.05 to 1.0 micrometers in diameter. Except for a few exceptions, mitochondrial DNA is maternally inherited. This kind of inheritance is called uniparental inheritance. It has been hypothesized that maternal inheritance of mitochondrial DNA is a consequence of gamete size. Egg cells are much larger than sperm cells, and thus, eggs are capable of holding more mitochondria. Another hypothesis to explain maternal uniparental inheritance of mitochondria in eukaryotes is that immediately post-fertilization, paternal mitochondria are selectively targeted for autophagy, whereas maternal mitochondria are not. Exceptions to uniparental inheritance of mitochondrial DNA includes paternal inheritance in bivalves and coniferous plants, as well as biparental inheritance in some flowering plant species. Moreover, the inheritance pattern of mitochondria is called cytoplasmic inheritance, which refers to the transmission of genes that occurs outside the nucleus.
Since mitochondrial DNA is inherited uniparentally, one might assume that a mother and all of her offspring would express the same mitochondrial diseases to the same degree. However, this is not always accurate because of a concept called replicative segregation. Replicative segregation refers to the change in proportions of mitochondrial DNA alleles as mitochondria replicate and segregate. A mother can express heteroplasmy, which would randomly result in her offspring being either homeoplasmic for wild-type mitochondrial DNA, homoplasmic for mutant mitochondrial DNA, or heteroplasmic for various proportions of both wild-type and mutant mitochondrial DNA. Offspring can inherit any of these different genotypes, and no two offspring have to necessarily share the same mitochondrial genotype. Inheritance is entirely random. In the homoplasmic wild-type mitochondrial DNA genotype, the resulting offspring express a normal phenotype. In the homoplasmic mutant mitochondrial DNA genotype, the resulting offspring express a mutant phenotype. And in the heteroplasmic mitochondrial DNA genotype, the resulting expression depends on the proportion of wild-type to mutant mitochondrial DNA the offspring possesses. Typically, when the wild-type mitochondrial DNA proportion is greater than the mutant mitochondrial DNA, the resulting phenotype is normal. When the mutant mitochondrial DNA proportion is greater, the resulting mutant phenotype is severe, and varying proportions in between depends on extent of normal to mutant phenotype expression. Consequently, the mother's offspring would express different levels of severity for the mitochondrial disease. Moreover, the likelihood of inheriting a mitochondrial DNA disorder increases throughout successive generations, since the successive rounds of replicative segregation results in greater proportions of mutant mitochondrial DNA if present in the maternal lineage.
Here is an example of what a pedigree reflecting cytoplasmic inheritance of a mitochondrial disorder looks like. This pedigree reflects a disorder called Leigh syndrome, in which 20% of diagnoses is due to mutations in the mitochondrial DNA that is inherited by subsequent generations via cytoplasmic inheritance. Leigh syndrome is a severe neurological disorder, and many of the mitochondrial DNA mutations associated with this disease disrupts assembly of protein complexes involved in oxidative phosphorylation, a critical process involved in ATP production that is carried out by mitochondria. The hallmark characteristic of this pedigree that affirms the inheritance pattern reflects mitochondrial cytoplasmic inheritance is due to the fact that the disorder is maternally inherited. As you can see, 2 passes the disorder onto all of her offspring, and only 5 and 9 pass the disorder onto their kids. 3 and 7 do not, since they are male and possess paternal mitochondrial DNA. Evidently, the mitochondrial DNA is being passed to successive generations via the egg from the mothers. Now that you've received some brief background, let's dive into some practice problems that illustrate some of the key points about the mitochondrial DNA.
Problem number 1 asks, 2 human populations have been isolated on islands since their ancestors first arrived. The mitochondrial DNAs of the people on one of the islands is more varied in sequence than the other. From this information, which island would you predict was populated earlier and why? The answer to question number 1 is that the more varied population is older because the mitochondrial DNA has had more time to accumulate mutations.
Problem number 2 involves this pedigree and asks, who in the pedigree has the same mitochondrial DNA as the 2-3 male? Give all correct answers. The answer to question 2 is persons 1-1, 2-1, and 2-4 because mitochondrial DNA is passed from the mother to all of her offspring. In other words, the 2-3 male will have the same mitochondrial DNA as his mother and all of his siblings as long as no replicative segregation is in play.
Problem number 3 asks, in a few organisms, traits encoded by mitochondrial DNA can be inherited from either parent. This observation indicates that in these organisms, A. Mitochondria do not exhibit replicative segregation. B. Heteroplasmy is present. C. Both sperm and egg contribute cytoplasm to the zygote. D. There are multiple copies of mitochondrial DNA in each cell. The answer to the question 3 is C. Both sperm and egg contribute to cytoplasm to the zygote.
Question number 4 asks, a physician examines a young man who has a progressive muscle disorder and visual abnormalities. A number of the patient's relatives have the same condition, as shown in the pedigree. The degree of expression of the trait is highly variable among members of the family. Some are only slightly affected, whereas others develop severe symptoms at an early age. The physician concludes that this disorder is due to a mutation in the mitochondrial genome. Do you agree with this physician's conclusion? Why or why not? Could the disorder be due to a mutation in a nuclear gene? Explain your reasoning. Here's the answer to question 4. The conclusion that the disorder is caused by a mutation in the mitochondrial genome is supported by the pedigree and by the observation of variable expression in affected members of the same family. The disorder is passed only from affected mothers to their male and female offspring. Additionally, when fathers are affected, none of their children have the trait, as seen in the children of row 2 number 2 and row 3 number 6. This outcome is expected of traits determined by mutations in the mitochondrial DNA because mitochondria are located in the cytoplasm and are usually inherited from a single parent. The trait cannot be X-linked recessive because in that case a cross between a female with 2 mutant X's and a male with a wild-type X would not produce daughters with 2 mutant X's, as we see in row 3 number 10, row 4 number 3, and row 4 number 4. It cannot be X-linked dominant because in that case row 2 number 2 and row 3 number 6 would have passed it on to their daughters, who are unaffected. The fact that some of the offspring of affected mothers do not show the trait and that expression varies from one person to another suggests that the affected family members are heteroplasmic with both mutant and wild-type mitochondria. Replicative segregation of mitochondria and meiosis may produce gametes having different proportions of mutant and wild-type mitochondrial DNA sequences, resulting in different degrees of phenotypic expression of the disorder among the offspring. Most likely, symptoms of the disorder develop when some minimum proportion of the mitochondria are mutant. Just by chance, some of the gametes produced by an affected mother can contain few mitochondria and result in offspring who lack the disorder. Another possible explanation for the disorder is that it results from an autosomal dominant allele. When an affected heterozygous person mates with an unaffected homozygous person, about half of the offspring are expected to have the trait, but just by chance, some affected parents will have no affected offspring. Affected individuals, row 2, number 2, and row 3, number 6 in the pedigree could have just happened to be male, and their sex could be unrelated to the mode of transmission. The variable expression could be explained by variable expressivity.